Hi, my name is Vivian Barry, and I am the lab manager of the Pathobiology team at Gilead Sciences in California. And today I'm happy to present a slide deck that I put together that talks about the principles of effective method development in pathobiology research. So here's an outline of what I'm going to be covering. I'll say a little bit of what the pathobiology team does at Gilead and the research that we supply. And then I'm going to go into several examples of using in situ hybridization assays on various projects. This is a very exciting assay that we like to use along with IHC, and it's been very impactful for a lot of projects. The first example I'm going to talk about is using a dual in situ hybridization and IHC assay. And I'll walk you through how we were able to identify a subset of B cells, in particular B regs. That's a very hot topic in tumor biology. And the second example will be how we used dual in situ hybridization assays. And I actually have three minor examples showing how it was impactful in finding a subset of macrophages that expressed a specific mRNA species. There were no suitable antibodies available for that protein, so I'll show you how we got around that. And second example will be identifying a subset of CD4 positive T cells. And the third example will be identifying a subset of CD8 positive T cells. So quite interesting and different projects and how this has been very impactful. And then last, I'd like to end with showing how we use standard in situ hybridization assays along with our IHC in times where we were not able to have antibodies that were available. We didn't have any robust antibodies for a good reagent for IHC. And also, we were able to identify proteins that have poor retention in FFPE. So I'll show a couple examples of that. So first, I want to talk about the overview of what we do at Gilead. We do many, many things, but I kind of summarize it into three important main tasks that we do. The first is target validation in human diseases. Now, anyone in the pharmaceutical industry knows that this is a critical step in drug development. We analyze human tissues to validate disease targets that will, in turn, support many decisions, such as a go or no-go for our therapeutic programs at Gilead. Target validation can help in finding better biomarkers in disease. It can help predict and measure disease progression and aid with patient stratification. So that's the first and foremost thing that we do within my team. The second thing we do is characterize and validate animal models. Researchers have assessed new therapies, mechanism of actions of therapeutics in animal models before assessing them in human subjects. So this has been going on. Many people are aware of this. But obviously, all animal studies can't be a direct relation to the human indication of interest. So characterizing and validating these animal models is essential to confirm that the animal model depicts similar pathology to the human disease, and obviously a well-characterized animal model can help predict the response in humans. So it's very important to validate these targets in the in vivo models before moving on to human tissues. The third and last component of our work in the pathobiology lab is evaluating drug effects, or we like to call it the MOA, the mechanism of action in models of disease. So animal studies can provide evidence of the effectiveness of a drug. We can assess if a therapeutic antibody or small molecule shows efficacy using these animal studies. So they help understand pathophysiological mechanisms of the drugs. We can determine whether it's toxic or if there's efficacy. And lastly, the data on the kinetics and PD markers or pharmacodynamic markers in animals can help aid in the selection of effective doses in the animal models and also going into human indications. So the first example of a method we've used in my lab is the dual staining with immunohistochemistry and chromogenic in situ hybridization, which I call CISH. It might be pronounced CISH, but I pronounce it CISH, just so on the next slide you'll see what I talk about. We use a dual IHC and CISH to detect a subset of B cells that express a specific secreted cytokine. Now, the B cell subpopulation I'm referring to are the B regs. They're now highly studied when looking at the suppressive role in the immune system in cancer, and it's a hot topic, definitely, nowadays. You don't just hear about T regs anymore, you hear about the B regs. Detecting cytokines with immunohistochemistry can be limiting, as cytokines are poorly quantitative, and actually they're very hard to detect since they are secreted proteins. And that's been an ongoing theme in my lab when trying to detect any secreted protein or secreted matrix enzymes. So we need to find a way to detect the localization of the cytokine of interest. So what we did was identify secreted cytokine C by detecting the mRNA by in situ hybridization. Commercial antibodies, I say here that they're not available for IHC. They actually were available, but they weren't specific. They were really hard to detect the secreted cytokine. So on the same slide, we wanted to follow with IHC using a membrane marker, CD19, to identify B cells. And so what we were hoping for, and what we did get, was that we were able to identify a subset of B cells, so they were CD19 positive, that also express mRNA of cytokine C. And I'll show you on the next slide, but what this was very helpful for us is that we were able to understand if these cell types played a role in cancer, these B regs in particular. We understood the population of these cell types in various human cancer tissues, and we were able to visualize the expression in spatial relation to specific cells within the tissue landscape. So we used the human tissue lymphoma as our positive control, and for our method development for IHC and CISH dual assay. And so first, the single-plex IHC that you see here showed nice and expected membranous staining of the B cell population in the lymphoma tissue. Lymphoma is filled with B cells, so that's why we use this as a positive control. Then the single-plex CISH showed the cells that were positive for the mRNA of cytokine C. So we had to optimize these conditions as single plexes first before we were able to combine and make it into a dual IHC in situ hybridization assay. And when we did the dual IHC, we could identify the B-reg cells that were of interest to our studies, and it was also a great way to help us understand if these B cells were present in specific tumor types in many different human cancers. So that was a very important assay that we developed in the lab. Now everything is automated, so it's much easier, but this is a great way to get around when you don't have any antibodies available to produce a robust assay. So the next example I'm going to show you is how we utilized a 2-plex in situ hybridization to detect 2 mRNA species and how they helped confirm data in our projects. In my first example, I'm going to show how we identify the co-expression of a protein and a macrophage marker by using the commonly used macrophage marker that everyone knows is CD68. We knew that protein A was expressed by macrophages. This was done through orthogonal methods, so it was confirmed through RT-PCR, Western blot analysis, but we were never able to visualize what subset of macrophage, if it was a subset or if it was all macrophages, if there were any that expressed protein A. We tried to find appropriate commercial antibodies for protein A, but they were not we weren't able to find good reagents. And so we were desperate to understand where protein A was expressed. We knew it was in macrophages, we didn't know where. And I'll show the images on the next slide, but you'll see again that we were able to identify key subsets of macrophages that were expressing protein A. And again, it helped redefine our understanding of the location of protein A in the tissue. So here, I'm showing that we used ulcerative colitis for our 2-plex in situ hybridization assay. We always run PBIB, which is on the bottom left-hand panel. This specific probe, PBIB, detects ubiquitin B, and we also have our negative control on the bottom right-hand panel. That detects mRNA of a species of bacteria, so that should not be detected in the human tissue. On the top panel, you see protein A single in situ hybridization assay on ulcerative colitis. As you know, ulcerative colitis is a very inflamed tissue. And here I have a zoomed in image of that, just showing you have that dotted line showing the border between the colonic epithelium and the lamina propria below. And here's a zoomed in image of that, where you see that protein A is detected in the immune cells below that green dotted line in the lamina propria, and also in the colonic epithelia on top of that green dotted line. We wanted to understand what immune cells, what immune-related cells were positive for protein A. So we did a dual in situ hybridization to detect the protein A. And first on the left, you can see CD68 IHC staining. If you have experience with colonic tissue, most people will know that the colonic epithelia can be sticky. But if you look at the macrophages that are located below the colonic epithelia, that's what we want to focus on. This marker is detecting the macrophages in the lamina propria. So there are many macrophages found in ulcerative colitis when you look at the IHC. On the right panel, you see protein A's mRNA detection by in situ hybridization. When we perform the dual in situ hybridization assay on the ulcerative colitis tissue, we could then visually see this co-expression of protein A's mRNA and CD68 mRNA. I'm going to zoom in so that you can see that. And I have an arrow showing a macrophage with protein A mRNA. So what was very interesting about this result is that we found that only the macrophages found near the lumen of the colonic tissue were expressing protein A mRNA. So this was truly a new discovery. Not all the macrophages were expressing protein A as we thought before, and it really was a subset of macrophages that we were able to visualize for the first time looking at the tissue architecture. So my next example is an example of using 2-plex in situ hybridization to detect 2 RNA species when we were looking for a subset of CD4 positive T cells, FOXP3 positive cells that are expressing a specific chemokine receptor. So a little background on the study. The specific T cells comprise a really low percentage of the population. I think we're looking at about 3 to 5% of the total CD4 positive cells found in human tissues. And we were expecting an increase when looking at diseased tissues, and we wanted to be able to detect this. The IHC reagents were available to test. We actually did have a nice, robust IHC assay, but we wanted to know if it was real staining. Because if we're looking at a 3 to 5% population of CD4 positive T cells, we wanted to be sure that we were looking or that we were getting the results that were true and specific. So in this case, we wanted to identify that the robust IHC reagent was truly specific and that it will also detect a chemokine, which we're going to call chemokine R, expressed in a subset of FOXP3 positive T cells. So here I have head and neck cancer, and you see FOXP3 IHC on the right panel. And what we wanted to do was, in that population, determine if any of those were FOXP3 and chemokine R positive. So again, we're looking at a population that's comprised of 3 to 5% of all the CD4 positive cells. And so what we determined is that if you look at FOXP3 in teal and chemokine R in red, there was a subpopulation of those FOXP3 positive cells that we could detect the chemokine R mRNA. Here's another example, another head and neck cancer tissue where we were able to identify a few double positive cells, again using teal for FOXP3 and chemokine R in red. And so where the arrows are pointing is a subpopulation of cells that were expressing chemokine R. And again, with all our controls, with all our assays, we have controls. On the left, we have our positive controls, and on the right, we have our DAT-B negative controls for the dual IHC assays. So my third example is to identify a subset of CD8 positive T cells expressing a specific chemokine receptor. So as you can see, there's a pattern here. Chemokines are secreted. We can't really detect them well with IHC reagents. So that's why we went to use chromogenic in situ hybridization. And this study, the background is that we were looking at chronic HBV viral infection in chimpanzees. We obviously don't do any research in chimpanzees anymore, so please note that these FFPE blocks were quite old, and we were still able to identify what we needed to identify using these blocks and also using the 2-plex in situ hybridization. So we wanted to examine if this specific subset of population of cells contributed to the viral replication when you have a chronic infection. So we ran the dual in situ hybridization in our control tissue, which we used was the cortex of the human lymph nodes. We wanted to make sure it worked in the human tissues before we ran it on the chimp tissues, which I mentioned were were processed many years ago. So we were able to identify a few double positive cells. These cytotoxic subset of cytotoxic T cells were identified. In the dark brown are the CD8 positive T cells, and then where you see red is a dual expression of CD8 and the chemokine receptor. So we were able to examine if this specific population of cells were affecting chronically infected chimpanzees with HBV. And so we were able to apply this onto the chimp tissues. So I want to get into other everyday applications that we like to use in situ hybridization. Just standard CISH assays, no dual assays or anything like that. And we basically use them almost on a weekly basis to validate antibodies, validate our reagents. And we also use them to identify proteins that either have poor retention in FFPE or where there's no antibody available to use for IHC purposes. So I want to talk about protein X. Protein X is a secreted matrix enzyme, and we knew it was highly expressed in colorectal carcinomas. And so typically, when you do IHC work, you work with FFPE blocks. And so on the left, we have protein X IHC. And if one were to look at this, you would think, well, it's not that highly expressed in this colorectal carcinoma tissue. It kind of looks like a murky stain, maybe even nonspecific staining. But this puzzled us because we knew that protein X was highly expressed in colorectal carcinoma. So what we did was we looked at the fresh frozen component of this tissue. This is not a serial section, but I will show you a few in the next couple slides. Then we got this result on the right, where we stained protein X antibody on fresh frozen colorectal carcinoma tissue. A very, very different staining pattern that you see here. We see a lot of staining in the stroma and no staining in the tumor cells. And we wanted to know, is this specific? Is this correct? And so the way we were able to answer that question is if you look on the left, there's IHC on fresh frozen tissues, and on the right, we did in situ hybridization on the fresh frozen tissues. The S stands for stroma. So just as we saw, these are serial sections. We were able to detect the mRNA of protein X in the stroma and little to none on the tumor cells. I think I have a zoomed in image here showing that these were actually activated fibroblasts that were making protein X, and it was actually secreted into the stromal component of the tissue, whereas this the tumor cells are almost completely negative. Here's the example I showed 2 slides ago. On the left was the IHC, and on the right is the in situ hybridization. So again, we were able to detect the stromal cells. These are activated fibroblasts. That's where the arrow is pointing to. And in this case, we saw that some tumor cells were slightly positive. So that blushy stain that you see on the left with the FFPE sample, that really was a true stain. It wasn't stickiness, but we couldn't confirm it until we saw the mRNA being detected by using in situ hybridization. So this is a close-up again showing the activated fibroblasts within that narrow stroma. The tumor cells close to that lumen where the L is, those are slightly positive. So what you see on the left with FFPE is a true signal, and we knew that we were actually detecting protein X by confirming through in situ hybridization. I like to show this picture because one thing about protein X that we know is that it's produced and secreted by activated fibroblasts, and these fibroblasts are activated more closely to the tumor stromal boundaries. So I have that depicted there by T:(S). We see the tumor cells with the T, and then we see the TS tumor stromal boundary. And if you were to look at an FFP image, which is on the left with IHC, it again looks like a very sticky, blushy stain. But when we did in situ hybridization, we were able to confirm that that is true expression of protein X at that tumor stromal boundary. So lastly, I like to show this example as well, where we had a really nice working, robust IHC assay for protein Z, and it worked very well in FFPE samples. The only problem, and I don't want to say it was a problem, but we didn't know this at the time, is that we truly thought protein Z was expressed only by immune-related cells. So we saw big reservoirs in macrophages, neutrophils. We never detected it in tumor epithelia until we stained lung squamous cell carcinoma, which is the example I have here. So we saw this very strong staining in the tumor cell epithelia, and we thought, we need to confirm this IHC because up until this point, we've only seen it in immune-related cells. And so we did do the in situ hybridization, and lo and behold, we were able to detect kind of a mirror image of what we saw with the IHC when you look at serial sections. And I have a zoomed-in image here. This was really nice to show that for the first time, we discovered that protein Z was actually being made by the tumor epithelia and not just by macrophages or neutrophils. This is what we normally saw with protein Z in the top right-hand panel, where you see the IHC. Protein Z was found in macrophages. Here we have tumor-associated macrophages surrounding this tumor island. And on the left, we have the in situ hybridization confirming that these macrophages, or TAMs, are truly our reservoirs of protein Z. And again, we have our positive control on the bottom left and our negative control on the bottom right. So this is the last part of my presentation. I just want to talk about, throughout this talk, we've talked about antibody validation, specificity, and we, with the validation steps that we do, we include orthogonal methods always. If possible, we'd like Western blot analysis. If Westerns aren't available, FACS is always good for surface proteins, ELISAs, and in situ hybridization, of course. And what we do is we usually start with cell pellets. We like cell pellets because you can use them as control reagents. You can overexpress, you could do knockout cell pellets. We work with a lot of phosphatase-specific antibodies, and so we're able to treat with phosphatase treatment to verify target phosphospecificity with cell pellets. So these are definitely reagents that we like to use and have when we do our antibody validation. So the way we go about it, and this was for a prostate target, we'll call it prostate target X. We make a cell microarray. We call them CMAs, not really TMAs. They're not tissues, so we call them CMAs, cell microarrays. And here we have an example of 10 different cell types. They all range in either being negative expression for prostate target X or medium expression, or high. And here it was nice, when we worked with the scientists, we were able to have an orthogonal method to compare when we validate our reagents. This is FACS analysis of protein X. And you can see that there are actually 3 cell types that are positive for protein X, and then you have 6 other cell types that are negative. And the positivity did range from low, medium, and high. So it's really nice to get that range of expression in order to do validation of your reagents. So when we tested several antibodies, we found the best one was this monoclonal antibody that seemed to adequately label the cell pellets. You can see the FACS data in the bottom right-hand corner of each panel, but the 3 positive cell types or cell lines that were deemed positive by FACS also checked out through IHC. This is a pretty clear go or no-go decision, but since then we've always incorporated in situ hybridization. So we went ahead and did the mRNA expression analysis by CISH on these cell pellets. And what was comforting and pleasing to see was that the CISH data corroborated with the IHC data that was produced, which also corroborated with the FACS data. So in summary, I hope I was able to convince you that using chromogenic in situ hybridization is a powerful technique to localize mRNA within fixed or fresh frozen tissues. One thing I didn't mention, but it's worth mentioning now, is when you use chromogenic in situ hybridization and not fluorescence, although fluorophores are much more stable nowadays, all you need for CISH is light microscopy. So these slides are archivable. You never lose their signal. And I've stained slides from 7, 10 years ago, and they're still bright with the chromogenic in situ hybridization signals. Using CISH or dual CISH or dual IHC and CISH, it really allows you to determine exact cell types involved. Not only does it help you identify cell types, but also subtypes of specific cells. We've used them as an adjunct to IHC assays, confirming ambiguous IHC results, or also confirming expression when we don't have a good IHC reagent. The data can be generated from both fresh frozen or FFPE tissues, where I showed you. And in the example where we had that secreted enzyme within the matrix, the IHC only worked in fresh frozen and not in FFPE, so we were able to make sure that that was a true signal. And we use this technique to validate reagents for IHC method development now. It's kind of like one of the steps that we, it's one of the check marks that we have to check when we do antibody validation. And so using all these methods have provided valuable and new insights to many programs at Gilead. And so it's always good to have orthogonal methods, but just being able to incorporate different assays to determine specificity is our #1 goal within the lab. So I'd like to acknowledge the folks in the non-clinical safety and pathobiology group, Chris Clark and Lori Deal, who are my managers, and Rosanna and Allison, who are instrumental in getting a lot of these assays up and running. Amanda Mickles-Vigdal was involved with a lot of the studies that I discussed today. And there are many folks at ACD, Advanced Cell Diagnostics, that have helped. Chris Bunker, Lee Fang, Shoban, Ruby, and the rest of the ACD support team. And the folks at Roche and Ventana. Without them, we wouldn't be able to get these assays up and running. So Judy, Wendy, Adrian, and Kelly, I appreciate their help and support.